So in topic 22, we're going to be talking about protein synthesis. Specifically, we're going to be talking about RNA, at least at this time. So genes, we know, code for DNA instructions. Basically, our DNA is a giant USB containing all instructions for whatever we need to make. How our hair texture is, how our skin color is, eye color, whether we have asthma or not, whether we have allergies or not, different things like that. It builds proteins that develop into us, whether they're working or not. So DNA contains those instructions. We got to decode those genetic instructions from DNA to RNA. There's a couple differences between DNA and RNA, but just like DNA, RNA is a nucleic acid that consists of long chains of nucleotides. It uses a base sequence that it copies over from DNA to direct the production of proteins. And then each nucleotide in both DNA and RNA are made up of 5 carbon sugar, a phosphate group, and a nitrogenous base. But there are 3 important differences. 1, the sugar in RNA is ribose instead of deoxyribose. 2, RNA is generally single-stranded, not double-stranded. And then RNA contains something called uracil instead of thymine. So there's no longer the nitrogenous base thymine. It's now called uracil. So here you could see the major differences. This side is DNA. So you have the double-stranded and you have the deoxyribose here along with the ribose here. RNA is single-stranded here. They both contain adenine, guanine, and cytosine. But like I said before, DNA contains thymine, RNA contains uracil. And now, remember how adenine used to be paired with thymine? Now it's paired with uracil. A good way to remember that is a pickup line that says, can I be adenine so I could be paired with you being uracil. So the DNA molecule stays in the nucleus because it's way too large to come out of it. So RNA molecules have to go into there to the protein building sites in the cytoplasm, which are the ribosomes. Ribosomes build proteins. So you can think of the RNA molecule like a disposable copy of a segment of DNA or a working copy of a single gene. So it usually finds whatever segment of DNA that specifically makes that protein, and it'll just get a small part of it. So RNA has a bunch of functions, but most of it is involved only in protein synthesis. So RNA controls the assembly of amino acids into proteins. Each type of RNA molecule will specialize in a different aspect. So we have 3 of them here. We have messenger RNA, also known as mRNA, ribosomal RNA, which is a ribosome. It's also called rRNA, and then transfer RNA, which is this one, which is also called tRNA. So most genes contain instructions for assembling the amino acids into proteins. The RNA molecules that carry copies of these instructions are known as mRNA. They carry information from DNA to other parts of the cell. Proteins that are assembled on the ribosomes, which remember they're located on the rough endoplasmic reticulum, or rough ER. So these ribosome subunits are made up of ribosomal RNA, and they can make up to 80 different types of proteins. And then once the protein is built, a transfer RNA molecule transfers each amino acid to the ribosome as it is specified specifically by the coded messages in mRNA. So there's 2 different parts. There's transcription, and translation. So I'm going to go over this first, and then I'm going to show you guys part of an Amoeba Sisters video in order for you guys to see it visually. So in transcription, segments of DNA serve as templates to produce complementary RNA molecules. So most of the work of making RNA takes place during transcription. So you can always think of transcription as transcribing. So transcribing usually means when you're writing something out. So imagine that the DNA has a coded message for you and you have to write it out, but it still doesn't make complete sense. And that's going to make sense in the next part. So during transcription, segments of DNA serve as templates to produce complementary RNA strands. So they're complementing them. Once again, not you're beautiful, more like it matches up. So A with U, G with C. And then the opposite goes as well. The base sequences of the transcribed RNA complement the bases of the sequence of DNA, of the segment of DNA. In prokaryotes, RNA synthesis and protein synthesis take place in the cytoplasm. In eukaryotes, it's a little more complicated. It goes from the cell's nucleus to the cytoplasm, to play a role in the production of proteins, so our focus is only going to be on eukaryotes. So transcription requires an enzyme known as RNA polymerase, very similar to DNA polymerase, and we know that DNA polymerase is a builder and it also is a proofreader. So we have something that's going to split it, more than likely, helicase, and then RNA polymerase here is going to build the mRNA. Notice it's a different color here. So it's going to build and proofread it. So RNA polymerase binds to DNA during transcription and separates the DNA strands. Then it's going to use one strand of DNA as a template, and then it's going to assemble the nucleotides into a complementary strand. Now, RNA polymerase binds only to promoters. So these are regions of DNA that have specific base sequences. Basically, there's a really bright sign in those DNA sequences that's saying, come here, this is where you want to start. Think of promoters at clubs or anything that's just like, come here, you can get whatever you want, just you got to come here. So promoters are basically doing that. They're signaling the DNA molecule to show RNA, there's signals on the DNA molecule that show RNA polymerase exactly where to begin making RNA. So similar signals in DNA cause transcription to stop when a new RNA molecule is completed. So once it stops, there's a certain way to read this. So remember how I said that DNA is coded and then RNA, you're writing it out. So you're writing out that code, but you have to write out that code a specific way. You basically write it out 3 letters at a time. Each word, in quotes, is 3 bases long and corresponds to 1 single amino acid. This 3-letter nucleotide code that you just made is called a codon. So we have to decode these messages to transcribe a nucleotide base from DNA to RNA. The transcribed information contains a code for making the proteins. Proteins are made up of amino acids that come together that form something called the polypeptide chain. There's 20 different amino acids. You can see this is one amino acid, and then this is a chain of them. So they start adding up together. So specific amino acids in a polypeptide and the order in which they're joined are determine which properties of different proteins. So like it could be something for your lungs, it could be something for your hair, it could be something for your blood, many different things. So the sequence of it influences the shape of the protein, which in turn determines its function. So RNA contains 4 different base types. Like I said, adenine, cytosine, guanine, and uracil. They form a language or genetic code with just those 4 letters. So each 3-letter word in mRNA is known as a codon. So here you have a codon. This is going to be towards a specific amino acid. This is another codon. Once again, going to be towards another specific amino acid, and same thing here. So a codon consists of 3 consecutive bases, and then they specify a single amino acid that's going to be a part of a polypeptide chain. Now, you're going to use this to read in order to, this is basically like a secret code, like how to unlock a secret code, a decoder ring. There we go. I couldn't remember the word. So this is like a decoder ring in order to help you understand what you just transcribed. You wrote it out, you still don't understand it. This is going to help you. So you start off here in the middle, in the biggest part. This is going to be the first letter. So the first one in the previous slide was AUG. So we start here with A. Then we go to the second part right here, U, and then G. And this tells us that it's this one, methionine. So that's 1. Now if I get another random one where I say GAU. So we have G, then we go to A, and then U, and we get aspartic acid. You can pause the video right now and try out different ones to see if you can understand how to take these steps. If not, just talk to me and Ms. Amayoa. We probably can help you. So a lot of them can be read on this. Some of them specify, a lot of them specify to the same ones. So like 6 different codons here specify to leucine, but that doesn't happen to a lot of them. So you have a bunch of them that specify to leucine that are here and here. But that doesn't happen too often. So methionine is usually known as the start one. So that's AUG. So whenever you want to start reading, that's what you look for. You look for AUG, and that's where you're going to start this whole protein. So ribosomes use the sequence of codons in mRNA to assemble the amino acids into polypeptide chains. And now we're coming to the part of translation. Remember, we transcribed it, we wrote it all out. Now we have to translate it. So the sequence of nucleotide bases in mRNA are a set of instructions that give the exact order of the amino acids and how they should make the chain. So the forming of a protein requires the folding of one or more of these polypeptide chains. Ribosomes use a sequence of codons in mRNA to assemble these. This process of decoding is known as translation. So messenger RNA is going to come out of the nucleus here. It's already got, you see here, the codons AUG, UUC, and AAA. So it's already been transcribed. Now we got to translate it. So it's going to go through the ribosome right here. And these are the tRNA. The dots right here are the amino acids. So the tRNA is going to come here. These parts right here that are opposite are known as anticodons because they're the opposite of the codon. So they're going to attach to the mRNA molecule, and as the ribosome reads it, it's going to direct tRNA to bring the specific amino acid to it, and then one at a time, the ribosome is going to attach each amino acid to a chain. So you're going to see in a second that they start attaching to each other. So this was originally the methionine one, which is the start codon, and it's going to leave to no longer has its dot, which is an amino acid. The dot is right here, and it's attaching to this one. Pretty soon this one's going to leave, and this dot's going to attach to this one. And that's going to continue on until it gets to a stop codon. And now we have a very long polypeptide chain that can be way longer than this. But this is a stop codon right here, so it already knows when to stop. So this is a smaller protein. So ribosomes are composed of 80 proteins and 3 or 4 different RNA molecules. These help the ribosomal proteins in place and help locate the beginning of the message. So they may carry out the chemical reaction that joins the amino acids together. Let me go and start the Amoeba Sisters video so then you guys could re-see this so hopefully you understand a little bit better.
In protein synthesis, we can look at 2 major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in, and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA.
This is known as the central dogma of molecular biology, and that information is transferred from DNA to RNA to protein. So most genes contain the instructions for assembling these proteins. You get a third review right now. So the DNA strand is here. The mRNA strand is going to come out to where it's being transcribed. Then it's going to be translated into the amino acids, forming a polypeptide chain, and therefore we have a protein. This is how genes are expressed. So expression doesn't mean like you make a face to it or anything like that. This is how it shows through in your body. So gene expression is the way DNA, RNA, and proteins are involved in putting genetic information into action in living cells. DNA carries information from specifying the traits of an organism. The cell uses the sequence of bases in DNA as a template to make it. So the codons specify the sequence of amino acids in a protein. Proteins play an important role. So now and then there are mistakes being made in copying the DNA. So you insert the wrong base, you skip a base, something happens, and these mutations can be inherited and change all genetic information. So there's two basic categories. There's those that are on a single gene, known as gene mutations, and there's some that are chromosomal ones that are on whole chromosomes, and they're known as chromosomal mutations. So mutations that involve changes in one or few nucleotides are known as point mutations because they're very specific. So they occur at a single point of the DNA sequence, and they usually occur when you're replicating DNA. If a gene is altered, the alteration can be passed on to every cell that develops from the original one. So point mutations like this, it can include substitutions, insertions, and deletions. With substitution, it only changes one amino acid. It's one letter changes, so it's no longer it goes from C to T, and that changes this letter, which then changes amino acid. With insertion, it shifts everything over. So you have this going here, so the A to U, the A and U both get inserted, shifts everything over. Now we have some kind of mess up. So all these amino acids change. Same thing with deletion. Now it all shifts this way, changing all these amino acids. Who knows what this amino acid will be now? So I just explained this. And this is also known as frame shift mutation because it's shifting the frame. So chromosomal mutations here involve changes in the number or structure of the chromosomes. So here we have the original chromosome. In deletion, we deleted B, the chromosome's a lot smaller. In duplication, same thing with B, now we have two B's, this became a whole lot longer. In inversion, they switch it, so B and C get switched here, so now we have ACB instead of ABC. Translocation is when you get, remember trans means change and location means obviously location. So this is coming from a completely different chromosome. So now it's getting inserted here and it's making it a whole lot longer and creating a different type of disorder. So these mutations can alter the natural events. Either this can be done by natural events or by artificial ones. So these mutations may or may not have an effect on the organism. Many mutations are produced by errors in genetic processes. So like when it's replicating DNA, that's when the point mutations usually happen. You got to remember that it's 10 million bases. There's way more than 10 million bases, but there's usually one mistake once in every 10 million bases. So small changes in DNA can gradually accumulate over time. Some mutations arise from mutagens. So basically this is when you're taking in chemicals or physical agents in the environment. So chemical mutagens like pesticides, few natural plant alkaloids, tobacco smoke, environmental pollutants, x-rays, and I mean like a lot of x-rays, electromagnetic radiation, and UV light, that's the sun, guys, by the way. These can all cause changes in your DNA. These mutagens interact with your DNA, and they can produce mutations at a faster rate. Others weaken the DNA strand, causing breaks, and these can cause chromosomal mutations. Cells can sometimes repair it, but sometimes they can't, so it can be changed permanently. So whether mutation is negative or beneficial depends on its DNA changes relative to the organism's situation. Mutations are often thought to be negative, but they actually can be pretty cool, and I'm going to show you guys something on a pretty cool one. So this woman is Elizabeth Taylor. She was an actress. She played Cleopatra in the past. So she actually had two cool mutations, and one of them is hard to see, but I'm sure you guys can kind of see how thick her eyelashes are. This is not mascara. She had a double row of eyelashes, which normally it can cause it to go backwards, and then you become blind. But she got lucky, and she just ended up with really thick eyelashes. Her other mutation is that she had violet colored eyes, which is like a bluish purple. It's hard to see in person, or at least in photos, it was easier to see in person. So, yeah, you can't, it's really difficult to see. No. But yeah, she had violet colored eyes, very gorgeous, cool mutations. So some of them, harmful ones, dramatically change your protein structure. These are produced by mutations that disrupt normal biological activities. Some cancers do that. Sickle cell disease is a disorder associated with the changes in the shape of red blood cells. I'm going to show you guys a photo that's going to explain this. So here's a normal DNA sequence. You have the DNA, it's going to get transferred here. This is still DNA, this is transferred to mRNA. We have this one right here. So this is a normal red blood cell. Then here with the mutant one, this is the one that's causing sickle cell. You have a change in this letter, which changes this letter, and then changes this, which causes valine, and that causes a sickle blood cell. So this sickle blood cell is what causes that weird shape, and therefore causes issues. But it's just one substitution that causes this. So there's multiple harmful effects. Normal blood cells being round, sickle cells appear long and pointed. These are caused by a point mutation that I just showed you guys. Remember that blood cells are the principal oxygen-carrying protein. So among symptoms for this disease are anemia, severe pain, frequent infections, stunted growth. There's multiple things that can happen with it. Some beneficial effects from mutations often produce proteins with new or altered functions. So some mutations have helped many insects resist chemical pesticides. Some have enabled microorganisms to adapt to new chemicals in the environment. And that's basically it. So it's not bad or it's not good, but it could be bad or it could be good, actually. So hope this helped.